brood for. There are deep sea octopus that have been known to brood for up to 54 months. That's 4+ years. So maybe that's the outlier, but we expect this to be at least, I don't know, a year or two that they're brooding, and we're just trying to learn more in every way. So what's happening with the water? What's the water chemistry like? What type of pollutants are down here? What is the temperatures that we observe in this water? Not by our crews, but by other research partners in the who've returned to the site since we discovered it last year. They have collected a male and a female from this area, so there are genetic analysis happening of the species to get to know a little more. We've observed hatchlings hatching out both during an Alvin cruise that Chad was a part of, and then also just earlier today we've seen about a dozen hatch from the site. So
all right, same thing, Aaron. Follow me in.
Lots observed and lots to learn, and a very long dive still ahead of us. So tons more time on the seafloor, tons more time to look around the area and see if we can find more octopus.
And did you mention that they are invariably associated with these kinds of vents?
I did not, but yes, we're not finding them just sort of everywhere. We're finding them in these pockets.
So you had mentioned that you haven't seen any juveniles. Would you expect them to be similar color, or do you really not know?
Yeah, the hatchlings were of similar color, actually kind of translucent. They're relatively small, obviously. They're just a couple of centimeters long, so you can almost see through their bodies, but they definitely have this grayish white color to them. There were a couple I've seen that almost had a red shade to them as well. So I wonder how variable that coloring is, but I do not know exactly what color they would be in their intermediate life stage. I would assume the white gray, but it'd be interesting to find out.
Come on.
So Chad, we have a few different sites now. We have Octopus Spa, and we have the Vent, and the original Octopus Garden. This is the site that Nautilus was at last year, right? This is the pocket?
This is not the site that Nautilus was at this year. This is the site that we discovered with the Alvin in March.
Oh, right.
But it was a more it was a better accessible site. The Octopus Garden has one decently sized pool, but it's a little bit harder to kind of land the ROV and conduct this kind of work. So this was a very optimal location because it's flat. Fantastic. And you can kind of navigate around these two pools, land in the periphery, and then kind of conduct your science.
Oh, so science, this one did not trigger.
Okay, well, we can try and shake it over a vent like you suggested earlier. If it fires, note that. If not, maybe it's just a dud, and we'll move on.
Let's see, there's a lot of fluid over this way.
I was happy to get, for Jeff, 2 samples at each site.
Video pushing?
And that's what we have. We have 2 at the vent and 2 here.
Would you like some grated Parmesan on your salad, sir?
Yeah.
Yeah, these are such neat looking samplers. Too bad this one's not firing.
Full wide video. That's pretty centered. If you try to
loosen your grip and
Yeah,
then
it's my
regrip.
next
Yeah.
goal.
So this is the only species of octopus that we've observed in this area. But you can see there are all kinds of other types of life down here. We have these really big anemones, some sea stars in view right now, crinoid in
There's
view.
some grip.
There's brittle stars and squat
Regripped.
lobsters and shrimp. So lots happening in the area. Sponge up in the upper right.
Maybe we just have a dud.
And those other types of animals would be the type of things you may be able to detect with the environmental DNA analysis that we'll do, in addition to knowing more about the bacterial population that's here.
Are we wrapped around the cable at all? The rope on it?
At the grip force, it shouldn't matter. We're wrapped around the rope on the other one.
Okay.
Yeah, so if this one doesn't look like it's going to fire, we don't have to waste too much time trying to get it to go.
Sure. All right, I'm going to put it back in the box.
Okay, we can get that in the funnel.
If we find something amazing, we can try it again on that.
So do you want a Niskin next?
Yes, and we normally, obviously, we kind of hover over these sites, but we don't want to disturb the mothers, so a Niskin in this position is just fine.
And we're going to put out these 2 temperature loggers.
Yes.
Or one of them?
Well, one of them right now, and then we'll save the other one for potentially at this site, maybe later today,
Okay.
if we find something new that we want to put it at. Maybe we'll find 12 degrees today, who knows?
So the difference between the squeeze samplers and the Niskins is just how specifically targeted they are. So the Niskins are mounted on the side of the ROV. And the water, they're cocked open with the tops propped, open on both the bottom and top of the tube, so that as the ROV flies around, water flushes through the tubes. And when you pull the triggering mechanism, when you pull
I'm going to go for the temperature logger next.
Yes.
We're going to see if we can get that little yellow one.
Yeah.
Yeah, when you go to trigger them, that snaps closed the top and bottom of the tube, trapping the water inside. But that is water that is over on the side of the ROV. In this case, where we see this shimmering and this venting, and it's coming up through the rocks, and we're trying to get as much of those concentrated chemicals as possible rising through the rocks, we have scientists and science team interested to get a sample as specifically as possible of those fluids rising up through the seafloor. So that's why we created that funnel. And by we, I mean our science team, and they brought all, really thought through these solutions.
Nice job, Greg.
Built this tunnel.
I don't know how you picked that up.
Yeah, that's crazy, Greg. So good.
Nope.
And then we're able to put that funnel right over the top.
But we can do
and put
this.
the intake for the squeeze samplers right into that funnel, so that we're getting that water, which we expect has a different chemical composition from its time, and will carry sort of signatures and clues as to what its journey underneath the rocks, underneath the sediment, has been like before it reemerges.
All right, confirming temperature loggers on the end. Aaron, let's push in. So,
Do you want me to brighten it up so you can see?
yeah, a little bit. I mean, this is kind of going to be a drop kind of placement.
Well, gonna have to dig for that one when you find it later. Are we marking not as a sample, or are we just
Not a sample,
but we are
but
marking the
a
location?
marker would be good.
All right. So it's right, that crevice that the logger is over is where we got the highest temperature.
Right.
And
Full wide.
is there a particular way we've been marking these?
And then we're doing a
I
Niskin?
don't know exactly what they did on the previous watch, but I was noting seafloor equipment deployed, and then I said temperature logger with yellow and white tape on handle.
Yellow.
So Chad, who's coming back for this and when?
Sometime next year.
Okay.
Okay, great. thank
Yellow
you.
temp logger deployed.
We will be able to make it out here in on some platform. We know that.
So then there's transfer
And we're going for 8.
navigation data so that you will know where to
correct,
look when you
8.
come back.
Yes, OET, last year's cruise, they just do a wonderful job here capturing all of the information for all samples, everything that's left out, all photographs, or frame grabs, I should say, and it's all presented in a nice little sleek James Bond type case of hard drives, and with a handcuff and everything, and I take it back to my office, and
The
I
football.
can, yeah,
Fire.
the football, exactly, and I can take it back to my office and
Nav, that's 008.
kind of do a search through keywords or look at a particular time in the transect, and make it very easy to find where things were left, where samples were collected with frame grabs of said sample or whatever. So, and then I can use those coordinates and come back, even if it's with another outfit or another ship. Yeah.
I mean, that's one of the most amazing things about OET, what they do. It's just everything is very professional and documented and easy to
you
navigate.
do a lot of arm work here.
Yep.
Just like not what you're used to.
It's a
That's
great
all
outfit.
the warm fuzzies. Thanks, Dane.
Yeah, absolutely.
Go ahead, Bridge. Whatever you want, George.
I'm pretty sure there's a recording somewhere.
Like looking for that missing shoe in the closet.
All right, so we are all stowed and ready to do whatever you want, Science.
Okay, so it was about 20 minutes ago, maybe a little bit longer. We saw those 2
that were walking across the
octopus that were walking around. I think it was a little further to the right. Let's see if they're still there. I don't know if they are.
Yep.
Yep. There's
Can I go check them out?
Yeah, I don't know if we have to swing the ROV over to the top.
Yeah, we need to spin around a little bit.
Okay.
Tether wraps, 0.7.
Okay, question. Viewers are interested to know if octopus closer to the vents are having more success with their broods than others, and we are just starting to tease apart, you know, some of the very basic questions. So we don't have a way right now. We're not leaving a lit camera that is going to, you know, specifically track them. We don't have a way to, like, individually, you know, I don't know, track or tattoo one, you know, group of octopus or put little trackers on them to know where they're headed after this. So
you can hold position here if that's possible. If we can zoom in on.
Yeah, Aaron, let's push in.
Just because the arm's out. So 1, 2, 3. This is a female.
how do you know that?
So do you see its right eye?
I I do.
That one arm that kind of extends down from that eye, if you will, is arm number 1. And then you just count clockwise from there, 2 and then number 3. And if you look at the tip of number 3, there's suckers all the way to the tip, and it does not have a modified end. Males have a modified end where it's called a heterocotylus. Well, they will take a sperm packet and deposit it in the female. So that is a female, and this one appears to be female as well. So the question is, what are they doing?
What you up to, lady?
Yeah.
Out for a walk?
And you know, this one here, it's interesting because kind of her oral cavity is face down.
Uh-huh.
I mean, it's possible she's looking for a place to lay eggs, but we haven't seen that process at all in the species, and I have no idea how long that would take, if it's a matter of hours or days. But they have a special gland where they produce a green cement, where they cement each egg one by one to the rock surface. So I'd presume that probably takes a while. I've seen clutches or mops of over 40 eggs for just one individual. So
Look at that shrimp hanging around.
Yeah, there's a couple.
Yeah.
They said that there was a predation on a baby octopus earlier by
I heard the
shrimp.
baby octopus got away though.
I heard
It was an
too.
attempted predation.
Yeah, wrestling match.
down in Dorado, they
They were hugging.
did witness shrimp when a female was moved out of the way. The shrimp came in and consumed the eggs fairly quickly. Here we haven't seen that yet, but yeah, I heard that that observation this morning, that once one hatchling came out and one of the shrimp grabbed it, and there was a little bit of a fight, but
A tussle.
a little tussle, but the baby octopus managed to escape.
All right, Aaron, let's come full line and figure out what we're doing next.
Okay, that's good for those two. That's good to know that they're female. Okay, so we can actually, yeah, float up a little bit, get a wider view, see if we can sit down somewhere else, get some more temperature and oxygen measurements.
Sure.
Wow, so the octopus kind of peter out there.
Yeah, there's two distinct pools here. One's slightly larger. They're both oblong, kind of elliptical. So you see the one on the left. You should see one on the right appearing soon.
Yep, it's just coming into view.
Yeah, so here's the smaller pool on the right as we float over it.
Wow.
This is a good perspective of the octopus spa here.
And the rocks look darker under
Yeah.
those pools.
Right, and that could be less sediment just
Yep.
from flow
So-
and octopus.
Let's see here. It may not be as warm, but I'm curious about the whelks again. So down to the lower right where you see all those snails.
Yep.
If we could sit down kind of in front of that, and we'll start taking a look around there.
Still part of the main pool though, like right in front of us.
Yeah.
You see a sea star. it looks
This is so incredible. Like, just to see.
Are we
this
going to be
pocket.
interested in the sea star, or can I land over the sea star?
We'll have opportunities to
Yeah.
collect sea stars
Okay.
later. That's fine. And Dane, I don't think we're brushing up too much sediment, so I think you're okay for the pump to be running.
Perfect.
Okay.
This is amazing.
So we've found not just this spa, but up on the top of this ridge, little collections of 15 to 20 octopus together. And what's really common is this whelk here. It's a type of snail, and we did collect it and get genetics run on it. It's sequenced, and it is a brand new species in terms of GenBank, meaning no one's ever sequenced it before. Doesn't mean it's a new species. Someone may have identified it 30 years ago, and just no one's ever sequenced it. But the photographs have been sent off to some whelk experts in Italy, so we're waiting to hear back. But there's a strong possibility that this is a newly discovered species of whelk.
So I'm not seeing whelks. What are we looking for here?
Oh, it's all on the bottom there, right above the porch.
I see.
The small snails there.
Oh, yeah, yeah.
So
Light cream color.
we presume this is one of the grazers in the area, kind of picking up the trash. I've seen them in a big clump consuming spent or empty egg cases. I'm not sure what else they perhaps consume. Sure, if eggs were available, they'd eat them too, but I'm sure the octopus guard them and protect them just fine. But in other little pools we found back in August with Mbari, this seemed to be the predominant grazer. And then we found one which there were hardly any whelks, and it was replaced by hundreds of sea stars as the predominant grazer. So these pools
They're
have
almost pushing.
individuality to them, and that creates even more questions. Why are the whelks not here, but the sea stars are here?
Well,
Yeah.
and look at them aggregated on the downslope edge of the octopus pool.
So interesting. Little whirls there.
Yeah.
It doesn't seem like there's a lot of flow around.
Yeah, I
The
didn't think this was going to be as much. Well, what we found is the octopus really do a pretty good job filling in all the spots with the highest flow rates.
Right.
And we have not been able to measure significantly high temperatures in places where there are not octopus. Maybe a degree or two, but nothing getting to 5, 6, 7, 8 degrees. So the octopus have done a pretty good job filling in every nook and cranny in the hottest spots. And which again arises the question that some of these areas that are a little outside are still pretty decent bare rock.
Yeah.
So then it's like, well, if that's still good bare rock, why aren't they there? So there may be something to the warmer water or chemical composition or something that's
Yeah.
attracting them. Maybe it just feels
Preferentially
good.
to the venting. Yeah, I mean,
you know,
I like warm.
I like it here in the jacuzzi.
Yeah.
it could be, it speeds up their development, so it could be an evolutionary advantageous adaptation
Yeah.
where they're able to turn over the population more quickly.
Right.
Maybe that's an
Absolutely.
advantage.
So we do know this species does experience senescence, so this is sort of the final stop. You know, they have a life history that means they lay these eggs, defend them, brood over them, keeping them oxygenated and safe for we don't know how long, but based on other species. And in the deep sea, it could be a year, it could be more. There are deep sea octopus that brood for 54 months. An individual has been recorded brooding that long, so that's 4 years long. And then this is the last stage. They are using up all of their fat reserves, lipid reserves. They do not eat for this time while they are brooding, and then they'll waste away. So reason to assume some of these shrimp would also be there as the recyclers, decomposers.
So I assume over the next hour or two at minimum, we'll kind of cruise around this whole spot and look for prime candidates to collect. We want to get some of the snails, one of those large anemones, a few other things as we see, but in the past it seems to be easiest to try and find loose small rocks with anemones or snails attached to them, because I know
Alright,
it could be
yeah,
problematic to...
We can slurp the snails.
Can you slurp the snails? Okay.
Yeah. And the smaller anemones we actually could probably do a slurp and scrape. But a larger anemone would be a grab.
Yeah, that'd be a grab on a rock.
I think a smaller one would be preferable because I heard a rock sample would be beneficial as well. I know we grabbed one already, but
Yeah, if we have eventually throughout the day grab a few rocks with anemones or whelks or whatever, then we get a 2-for-1 sort of. We're not planning to collect too many rocks. We're not trying to load up a lot of weight.
All the
on
rocks.
porch.
Got it.
All the rocks.
Look at the amazing behavior of that one in the center, and the one upper left, kind of coiling the ends of their arms repeatedly. What is going on there?
Yeah, kind of like that. I've heard it called the egg beater. We've seen lots of octopus do that, where they kind of swirl them like that.
Formulating their dastardly evil plan.
Wishing they had a mustache to spin.
So yeah, if we can focus in on that one in the upper left, there are eye spots there, so it'd be good to get some frame grabs, see if we can see arms too, which we probably will.
No, hey to Friday Harbor High School oceanography class. Cool to have you with us.
All right, Aaron, work your magic.
There's just one egg per egg case. And some individuals have been seen up to 40 egg cases. Is that right, Chad?
Yeah, that's about and I'm sure there's more than that, but that's been one where we were able to positively count. The problem with these moms is they're very good at hiding their eggs. So you often cannot see even a couple, but if you can see, like for example right here, we can see eye spots and developed embryos, but we can't really determine how many eggs she has.
Sure. Chad, lots of people want to know. They're shocked and you know, in a little disbelief that they don't actually eat during this time. So how do we know that they don't eat? How do we know they don't like leave and then come back?
Right. It's just very common in octopuses. This is what they do. They lay their eggs and they're done. They don't go off and eat. And if they were to go off and eat, they leave their eggs exposed anyway, and that's not something they're going to do. So it's not something that's been witnessed, as far as I know, in my knowledge, that moms have said, "Hey kids, just stay here. I'm going to go grab a bite to eat and come back." This is their mission in life. Once they get to this life stage, they lay their eggs and brood them until they hatch. Oh, there's some more developed embryos there.
That's so cool. We know they're developed based on that dark spot in there. That's an eye spot developing. You
Yeah.
can see through that egg case. It's really cool.
And after we discovered this place in October, and we went back out with the Alvin in March, they asked me, well, how do you know they're going to still be there? And that was a really good question. So it was just kind of an educated guess that there's probably a revolving door here.
Unfortunately, not really.
These mothers probably take a couple of years to brood, so it was reasonable to assume that, you know, a quarter of the population maybe just laid their eggs, another quarter or half are in the intermediate stages, and another quarter are getting to the end, ready to hatch. And so when mothers senesce and die off and move on, a new mother comes into that spot. And so there's just this perpetual population here. There's not a mass migration in and exodus out, like a coral spawning event or something like that.
Which does tend to make a lot of sense, just because you're in a deep sea environment, which does not change. It doesn't have seasons per se. So they could be able to have kind of ongoing reproductive cycles at different different stages, different times a year, just to kind of support that theory.
Sorry, I couldn't hear that.
Do you want to start with our temperature probe or with the temperature oxygen combo probe?
Either one. I guess we can start with the Herc's probe.
All right.
We have a couple different tools here that we're going to use to measure the temperature, just to compare the tools against one another and also just gain more information about this area. So we have Hercules' temperature probe, which is, if you're watching the channel 1, has a round ball handle on the top and a long pointy metal rod extending out the front, holding the octode. And then we also, or the temperature probe, and then we also have that larger white, gray, and black octode, which is a combination oxygen concentration and temperature probe. So we're going to try with both.
Oh, our companion maybe.
Video. Let's. You already. Nope. We're grabbing back on.
Nope.
I thought he was going to come off.
She's staying.
Or she.
Nope. I had high hopes there for a moment.
Hello. Yes.
You're not Megan. Is it still Megan? Where'd Sam go?
Perfect.
Can't hear you. Oh, got it.
Can everyone hear me?
No.
I
No?
can.
How about now?
Yeah, better.
Better. Strange. Stand by.
Oh, there
Your
you go.
volume seems right. Yeah. If you test it again,
Testing, testing, 1, 2.
Yeah.
yeah, you're peaking
Loud,
correctly.
Okay. So were you hoping for a round of introductions now that your actual science communication fellow has left?
Yep.
Perfect timing. Sorry, Megan. I'll pretend to be Megan. Is now a good time?
Sure.
Yeah.
What are we introducing?
Well, ooh, okay, now that I'm here, let's see.
And then
Uh,
we're getting
How about
technical.
name?
affiliation and role on the ship, and favorite octopus you've seen today. Just kidding. How about
all of them?
how about favorite ocean animal?
Oh.
We'll start with Erin.
Oh. My name's Erin Rainey,
All right, video, let's push in a little
and
bit.
I'm pushing in.
Hold that.
I'm the video engineer on this 8 to 12 watch.
Lines flow.
I am a freelance wildlife camera woman, so no affiliation. And my favorite ocean creature, I'd say salmon. Your turn,
Excellent.
Paul.
Hi everyone, my name is Paul. I'm the ROV engineering intern. I'm helping
Right in
fly
the shadow.
Argus. My favorite animal is the flamboyant squidworm.
Very nice, Paul. It's a good one. It's a good one
Aaron,
to
let's
choose.
brighten that up a little bit. Thanks.
Is that good? Do you still need it bright?
And Paul, where are you coming from?
Oh, San Diego, California.
What? Where are you?
Or
Doing your
UC San Diego is
Perfect.
grad school.
Come on, Paul.
Okay, my name is Greg Diffendale. I'm coming from Florida and Clearwater specifically, and I'm your Herc pilot. My favorite underwater creature is that coppery bristleworm.
Oh yeah.
Oh, that was
really cool.
really cool.
Was that today?
No.
No, I've only seen one this year. You don't see them all the time.
It's really like
Where did you see it?
metallic in Cordell Bank or Greater Fairlands, one of the two.
Cool.
Well, if the nav doesn't know where we were, I don't know where we were.
Speaking of nav,
Hello, Renato Kane, navigator on this watch. I'm from Wilmington, Delaware. My favorite deep sea animal is the deep staria jelly.
Oh,
Ooh,
he has
and-
evolved and moved on.
Right.
It's because everyone keeps
call.
taking his favorite.
the squidworm.
If someone
Uh,
starts to take this one, I'm going to
I
be
think
upset.
I'm going to, because
Yeah, I
I
like
got
deep
to
story.
film that.
She's the one who filmed it, right?
That's
So
true.
she has rights on that one.
Well, Rennie, do you like deep staria from the ROV Hercules archipelago or the deep staria from
Ooh.
Just
Baker?
recently?
I'll be
Bigger
honest,
Oliver
I
Johnson.
like the most recent one better.
Oh, ouch.
Ooh, with the resident isopod.
They both had that.
Excellent. Well, folks at home can check out photos and videos to see both and compare side by side to
Yeah.
deep staria.
I like the Mexican one.
Me too.
The one in Mexico, the isopod was white, and the one Howland Baker, it was red.
Well, we're getting pretty good temperature here.
Yeah, almost 8.
All right, let's take it to the back row.
Jamie.
Hello, I'm Jamie. I'm the data logger on this watch. I'm a contractor with OET and act as science manager on board. I'm from North Carolina, and as it happens, my favorite deep sea animal is octopuses, so I'm very happy.
Yay!
It's just the best day ever for
It's the
Jamie.
best
Well,
day
explain
ever.
to us why you were
Yeah.
an anglerfish last Halloween.
Because that's also a cool animal, and you
Okay.
can't be an octopus all the time.
Yeah, she's got to mix
Unless
it
you're
up.
an octopus, in which
Yeah, I was
case you
gonna say
can
they
be an
can.
octopus
Yeah.
all the
They
time.
seem to be doing a good job at it.
Profound words. Jamie, do you have a specific species that is your favorite?
It's too
Oh.
hard to pick.
It's okay. Take your
This
time.
one's really great, though, I have to say.
The moose octopus.
Moose octopus. I like its name, too.
Yeah.
But
I like
yeah,
the antlers.
I don't know if I could pick a favorite. That would be really hard.
9.3.
Whoa.
Killing it.
Off the charts. Well, not really. The charts are adjusting to our temperature, so we can't actually be off them.
We can try.
We can try.
Amity, you're up.
Yeah. My name is Amity Wood from Santa Cruz, California. Woohoo. Work for Monterey Bay National Marine Sanctuary as an education coordinator, outreach coordinator. And gosh, I don't know, everyone's taking the really cool critters.
Video, let's push in all
I
the way.
might have to go for
and
some
You're good on bubble.
charismatic megafauna, so I'm going to I'm going to pick Pacific leatherback turtle.
Ah,
Nice.
Yeah.
Nice.
Yeah.
That's a good one.
Good range on that one.
Yeah.
We don't get those in Florida.
On account of them, the Pacific title.
Yeah.
Yeah.
That's it. Yeah.
Well, we have orcas in the Gulf.
Last but not least, or second to last but not least, Chad.
yeah, Chad King, hailing from Gilroy, California, and work for Monterey Bay National Marine Sanctuary with Amity. I'm a research specialist on the research team and lead scientist on this cruise. And favorite animal, I mean, look, this one here that we discovered last year is kind of way up there. But during my career, I've always had an inkling for the wolf eel. They look like an old grumpy grandpa.
Yeah.
And I just love their disposition.
Those are incredible.
And the fact
looking.
that they mate for life and take care of their eggs. So
Yeah.
pretty cool.
Who couldn't
And-
love that face, too?
I know. I just want to pinch the cheeks there.
And Chad, you've had a long career as a scuba diver, and have you ever encountered them underwater?
Yes,
And-
absolutely.
Have
Any
you actually
unusual
pinched the
encounters?
cheeks?
Yeah.
Is I think the real
I
question.
might not have a finger after that. No,
Yeah,
I have not
that
done
seems
that.
like a
But
dangerous idea.
I do feeding shows for Monterey Bay Aquarium, and we have a wolf eel in there. So I routinely go back and hand deliver squid to him. He used to come out all the way to the window. I would coax him out with some squid, and he would come all the way out, and the crowd would ooh and ah. But he's truly like a grumpy grandfather, get off my lawn type now. And he says, I know you're going to drop it. I know you're going to drop it. And so I oblige him, but I love that animal. And we got up to 10
Well, I was
degrees.
going to say, we just hit 10.
Wow.
Wow.
Okay, so that's a new
Double
high
digits.
Yep.
for this spot, and only 0.4 of a degree below the highest recording we've ever got in this entire neighborhood,
Wow.
which we got in August. We got 10.4.
All right, the spot is heating up.
Video, let's come wide. There was one egg casing under that octopus that looked like it was empty.
Yeah, I saw that.
Eagle-eyed Jamie in the back. All right, let's push back in video.
Is that probe as far as it can go pretty much into that crack?
That was my next goal was to see if we could actually beat the other record.
Wordy at 10. That extra
Aaron,
inch.
push in more. And we're going to need to brighten. Oh, it's right behind the, okay. No, whatever we did was wrong. Full wide.
That's okay. So you can either try and readjust, or we can just grab the other temperature logger for comparison. And we don't have to take too many temperature measurements here. We just wanted to get an idea, kind of the range here, and then
Right.
get some baseline oxygen readings, and then maybe do some collections before
Video
moving
pushing.
on.
Let's see. No, that was kind of our best flow spot.
Yeah, a lot of times the dark rock really indicates where the flow's coming from.
Thanks, Paul.
So how long have we been leaving the temperature probe in at each site?
Till it gets to the max.
Okay, so less of a time specification and more of a,
Yeah, if
Wait and
it's
see.
if it stops trending up.
got it.
Boy, that one's not the right spot.
Yeah, that looks shimmery right there.
Chase the shrimp.
It's just hard to figure out where the shimmer is, if it's in the foreground or the background.
Yeah, the depth perception of where it's
Right.
coming from it's hard to assess.
That also seems like not a great spot.
Slowly climbing.
Slowly, yeah.
All right.
The other one took a while to get up there, so
But usually you can get to 5 pretty quick.
yeah.
There we go.
There it is.
Perfect.
Right,
Yeah.
see that.
So while we're waiting, we have Dane in the back.
Yes, hello everybody. So I am Dane Harden, also from Santa Cruz. So Amity and I are a couple of people who help keep Santa Cruz weird. We compete with
Hey,
Austin,
speak
Texas.
for yourself.
We had it first. I will speak for myself. I am with Applied Marine Sciences, and we are a consulting company. We've operated a regional water quality monitoring program in Monterey Bay for the last 18 or 19 years. And I am on board to collect water samples using specialized sampling media in which we will analyze legacy contaminants, so things like PCBs, DDTs, chlordanes, flame retardants. And this is part of the sanctuary's efforts to understand
We're on
human
track for a record
impacts
right now.
on resources in the sanctuary and to protect those resources. And we have periodically seen very high concentrations of PCBs and DDTs in surface waters of Monterey Bay, and which we suspect are coming from land, but fairly inconsistent in the signature and concentration. So collecting these very deep ocean samples is another way in which we hope to understand the
That's what I'm
sources
watching.
of these coming into Monterey Bay.
So we are
Oh.
approaching. Sorry, you can
Yeah,
tell your animal.
my
here.
favorite animal is one that is found mostly in shallow waters, sea squirts. And I love them
Ascidians.
because, yes, ascidians, tunicates, whatever you want to call them, sea squirts, they are urochordates, so they're in the superphylum Chordata, of which we belong, and they are there only by virtue of their larval phase, which has a very rudimentary notochord in it. But when they pass out of the larval phase, they settle down, they stick to rock, and turn into filter feeders.
Well, you guys have found a good spot. You've matched the highest temperature we found in the entire octopus garden.
Wow.
Wow.
10.4 degrees Celsius. So maybe we'll let it sit here, see if we can kick it to 0.5.
Awesome.
It looks like it might be leveling off here.
Yeah, we're at 10.40, 10.39. But it's a good confirmation that it's the same temperature, or that you can find the same temperature.
Yeah, I thought that that vent might be the highest we find for a long time, but you guys got a nice rubble here that you can get the wand in, so it's less mixing with bottom water.
Was that with the same probe?
No, it isn't with the same probe, so, you know, obviously, assuming they're both calibrated
Yep.
correctly, it should be comparable, but at least it's reasonable to say, because, you know, there's already a little bit of an art to this anyway.
Yep.
We're not...
They're both Hui temp probes.
Oh,
Right.
there you go. But, yeah, depends. You can get lucky and
This is
find a
pretty
conduit.
solid,
This is
10.4.
pretty solid, stable.
Yeah, no, that's great.
Oh, 10.41, yes.
Which we can't see on the screen.
Yeah. We've got one more sig fig than you guys.
Yeah, so for those of you listening, this is
So this
in
seems
the low
pretty constant.
50s.
I'm going to try a little wiggle here, but otherwise we'll grab the other probe.
Yes,
and stick
that'd
it
be
here.
great. Give it a wiggle, see if we can get it bumped up a little bit, and then get the other probe.
So low 50s, that's like normal surface temperature in Monterey Bay.
Yeah,
Yep.
yeah, it's
Area.
good old scuba diving. You still need a wetsuit if you were down here.
Yeah.
Never mind the pressure and everything
Yes.
else.
Some other
Never mind, you'd be
considerations.
crushed by
Yeah.
the weight.
Yeah, right.
So what is the normal temperature down here then normally?
It's been about 1.7, 1.8.
Okay. It's quite a big bump.
Which
It
in
is.
Fahrenheit is
So when we say warm, it is-
mid-30s.
Yeah, it's 34,
Yeah.
35,
Yeah.
somewhere around there.
Yeah.
Yeah.
It's
No,
very
we are,
close to
we are
freezing.
still solid 10.39, 10.4.
Awesome.
All right,
The sweet spot.
let's come full wide.
Yeah, right now it's about 1.6 degrees Celsius in ambient bottom water right now. So that's almost a full 9 degrees warmer.
Wow.
Centigrade.
Yep.
Yep.
Is that a crinoid to the bottom right underneath the predator arm?
Bottom. No, that is a black coral.
Black
Oh,
coral.
interesting.
Just checking what that
Oh yeah,
Yeah.
I see it.
Uh,
It looked like the large stock crinoids we were seeing a lot in the Central Pacific, but we are no longer in the Central Pacific.
go on to your, you want to look back in and see if the hose is feeding properly. Oh, mostly properly.
A loose interpretation.
Likes the wrist.
You can see it well on bubble.
Oh, good. Awesome. Come back out here.
Oh yeah, there you go.
Yeah, the trick is to not get any of the cables. Okay, video, let's push in. Hold that. Right, and so then a lot of this is either shadow or touch, right? So I know I'm not going there. But I want to bring all right, full wide video.
A lot of assistance today from the
Yep.
octopus.
And then I'm going to come under here and
So which tool do we have coming out now?
this is a live oxygen temperature readout. Full wide. Oh, video, you're already full wide. Thank you. I have to do things. So oftentimes when you grab a T-handle, you'll end up with it in that little kind of square.
Mm-hmm.
In this case, for what we're doing, it's a fine grip. But that's what they're actually designed for.
Is this grip?
Not the grip that I'm in now, but
Oh, in the square, right.
yeah.
Don't you have more dexterity with it out where it is now?
Yeah, you can kind of, I can put the end on a rock and rotate exactly how it's in the jaws, so depends if you're doing like something that's more forceful versus...
Right, right. Like driving it in.
Correct.
So Chad, on the previous cruises to this site, were you also testing for dissolved oxygen levels?
We do have oxygen readings from August, but they were not interpreted yet. So we have kind of the
Video
raw
that's
data.
pushing.
Okay.
But the benefit of today is this is...
Hold that. Where exactly was I
I'm
last
looking
time?
for the term, but basically it reads in the
I think
proper
I was
unit.
right
So we
there.
kind of know relative to bottom seawater what the percentage of oxygen is.
Got it.
And earlier today they were getting oxygen levels at only 50% of ambient seawater. Which again, the question arises, well, these
Yeah, I
eggs
think
need
it was
oxygen
that one.
to grow and develop. What's their threshold where it's too little? Because down in the Dorado outcrop, they were finding that the higher the temperature, the lower the oxygen was, and they didn't find any development in their eggs, and certainly no hatchlings. Well, here we find development all over the place. We found tons of hatchlings.
Like that PVC
yet
tube might
the
just
oxygen
be
has been
hard
as low
on
as
that
50%.
rock.
So
Yeah, I mean, there's
why are
a
they
little bit
surviving
of
here
flow, but there's a
differently
crack
than the Dorado?
that
We
should
don't
be
know.
kind of over.
Is it not on right now, this optode?
It is no data points.
Yeah, it says no data points.
Interesting. No, ours is receiving power, so it has power.
Herc, do you think Justin would be any assistance in this instance? Should I try to call him or no?
It would either be Justin or Annie.
Okay.
I don't know how to use their little
Okay.
laptop.
Let me see what I can do.
Yeah, I think Annie's asleep because she's on 12 to 4 and she was up late, so maybe we start with Justin.
So we got a lot of folks just joining us now here in the Octopus Garden, where we are doing some temperature and oxygen
Oh no.
readings along these
There
shimmery fluid seeps here. A couple of questions people have asked are, what are the orange things in
I
between
can
the
try to
octopus?
just turn it off and turn it back on again.
Yeah.
Those orange things are anemones growing on the rocks
Yeah,
here.
so Herc, the previous watch contributed that they cycled power
Yeah,
and
that's
that
what I was gonna
worked.
do.
Okay, cool.
Let's see. Off. And on. It's got a little boot up sequence it goes through, if I remember correctly.
So behind this oxygen sensor, you can see one of the green lasers. There's another laser spot on the sensor right now, but those are scaling lasers up to 10 centimeters. So we can measure distances underwater or the size of organisms underwater. There they are, thanks. In this case, you know, it looks like a lot of these octopus are just over 10 centimeters.
There it is. Came back online.
There we go.
Nice.
All right, I want to confirm with everybody that it was right behind. Video, let's come wide. Let's see, that it was right behind, I believe, this octopus that we got our readings. Because we weren't stretched out very far, and it was an octopus that was kind of fully exposed. Not
Yeah, you were
sideways.
able to get that other probe in between the rocks. So I would
Yeah.
try there first, and then a secondary one would be to the lower right in that dark rock area.
Yeah.
All right.
So let's get 2 readings here.
All right, video, let's push back in. We're gonna get as close as we can without plugging up the tube. Hold that.
We're starting to get a little bit of a temperature increase. Oh yeah. Oh no, the scale's not that big, but
Oh, vehicle, what you doing?
so I might hold it. I know the web page says it's refreshing every second, but I'm not seeing new data show
Yeah,
up that
there's new
often.
data that actually, it pipes up new data every 5 seconds. Oh, there's a little rock with an enemy on it.
Yeah, I saw that too. If the later when we collect, that might be a candidate.
Full wide for a moment, video. Thanks. We're going to do... All right, let's push back in.
There's a nice shot of the eggs there.
Yeah, nowhere near the same temperature readings.
Yeah, I just can't get to that flow.
When's the last time it was cleaned? Yeah, right. When we were in the med and we were allowed off the boat at times, it was every 2 weeks we would check it, clean it, and put it with, coat it with some grease to prevent biofouling.
Yeah, the symptoms don't seem to,
Lend themselves to that.
yeah, just because of the intermittent, it just cuts out and then.
This is about as wiggled as I can get this in there without really starting moving rocks around.
Yeah, no problem. Leave it
I'm
here for a
also
little bit.
capable of moving rocks around, so.
So that hole in the white there is where the outflow
Yeah.
goes through. So it looks like it's sheltered from the current.
Mm-hmm. It just cools down physically, but
Yeah.
in that transfer up the tube.